continue a discussion the clinical trial. So the last part that we discussed in the last lectures is the comparison between the superior trial and the non-inferior trial. So we have other types of a trial that we'll discuss here. So this superior trial demonstrates that the effect size of a test drug is superior to comparator. Comparator like existing drug, that's we comparing between two drugs like the tested drug and also the existing drug. But the superior, that's mean the new drug or the tested drug, it has more effect or better effect than the existing one, while the non-inferior, that the new drug is not superior to the past drug or existing drug. It's not worse, it's good, but it will be the same effect maybe to the existing drug. And that is the difference between the superior trial and the inferior trial. So again, the superior trial demonstrate that the effect size, and we can see here from the drawing that we can show in here, demonstrate that the effect size of the tested drug is superior to comparator. That's something that we compare to. And the non-inferior trial demonstrate that the effect size of the tested drug is not superior to the comparator. It will be good as well as the existing drug. And the other topic is, what is the protocol that we do it. It has objectives, it has design, it has methodology, it has a statistical consideration, and the organization. These are what we call them as a protocol. So the protocol, it has objectives, it has design, it has methodology, statistical consideration, and organization. So again, about the drug development, and we discussed that again, the preclinical development and then the clinical development and then the post approval. The preclinical, actually it composed of the two stages like discovery, and we call them as a research. And the preclinical that we do it in animals, we study the pharmacology and toxicology in the animals. And phase 1, it will be in the human, in the volunteers, in a healthy one. And the phase 2, it will be first in the patient, and phase 3, it's a proof of the efficacy and safety, and phase 4 is evaluation in the real world, in the real clinical settings. And here what we want to study, what is the objectives, and we said that the protocol, the first thing is important is the objectives of this protocol. So the phase 1 of a clinical, it's a human pharmacology, as we said. It's the phase 1, therapeutic exploratory phase 2, therapeutic confirmatory phase 3, therapeutic use is phase 4. So we will start about the phase 1, which is a human pharmacology. The objectives, because it happens to the volunteers with they are healthy one, so assesses the tolerance, that's mean how they are pure of these effects of the drug, and define, describe the pharmacokinetics and pharmacodynamics of the drug, explore a drug metabolism and the drug interaction, estimate the activity. So this is that is happening in the phase 1, which we can call it as a human pharmacology, because it assists the tolerance. We just explore things about explore the activity of the drugs, the interaction of the drugs, the pharmacodynamic, the pharmacokinetics of the drug, the dose of it, the tolerance of the human. So that why it's happened in the human, in the volunteers, in a human, in a healthy one. And the study example like dose tolerance study, like maybe we study it in the healthy one to explore what is the maximum dose that we can give into the human and not causes any toxic and any effects, bad effects on the human, like dose tolerance studies or single and multiple doses, pharmacokinetics, pharmacodynamics studies, and the drug interaction. So again, human pharmacology is a phase 1, it given to the volunteers, and we said before that is from 10 to the 100 volunteers that we use them in this phase. And actually we wanted to know what is the tolerance, what is the drug use, dose, and we wanted to know what is a pharmacokinetics, thermodynamics, interactions of the drug, and these things, And then we go to the other phase, which is phase 2, which is done as therapeutic exploratory. This phase we do actually to the real patient, and the number of them may be 100 to the 500 patients. And the objectives of the study: explore the use for the targeted indication, estimate the dosage for subsequent studies, provide basis of confirmatory study design, endpoints, and methodologies. We do that now to the patients, and now we can see what is the effect. So the first thing that he said, explore the use for the targeted indication. We just discovered these molecules or these drugs, and these molecules, how they are interacted with the target. And I mean a target, it may be receptors, it may be enzymes, it may be protein, whatever is it, and we know the interaction between them and how that is affected it, and what is the dose that is important for this target, and how much the target need for these molecules to be interacted, these things. That's why we call it exploratory, because we discover things when we are dealing with a patient. The other thing, estimate the dosage for the subsequent studies. Actually, when we are studying what are the target indications, we can now know what are the dosage form, and we provide the basis for the next step or for the next phase, which is confirmatory therapeutic. And the study examples is the earliest trials of relatively short duration and well-defined narrow patient population using surrogate, and we understand what is surrogate now. What is the surrogate endpoints? We study the surrogate endpoints, and we said that and the pharmacological endpoints for the clinical measures, and we have do exactly the dose response exploration study. So these are the examples of the therapeutic exploratory. The first one is the earliest trials, because we do that in the patients of relatively short duration in a well-defined narrow patient population, and we said from 100 to the 500, in using surrogate. And we said that surrogate is just predicting the real, we can say that the predicting what is happening in the real patients, like we making a comparisons between the cancers and the tumor size and how it's reduced, and that's mean it's more survival. It's not sure, but it's the surrogate endpoints, and the pharmacological endpoints for clinical measures. And we do also the dose response exploration studies. It's about explore things, and then the therapeutic confirmatory. This is the phase 3. Demonstrate, confirm efficacy. When you just hear the therapeutic confirmatory, all that happens to your mind is efficacy and safety. Demonstrate, confirm efficacy, how it's effective, this drug, how that is make a big effect to the patients. And in the same time, establish safety profile. There is like reduced side effects, and it's safe for the patients. Provide adequate basis for assessing the benefit-risk relationship to support licensing, and then establish dose response relationship. We know what is, like we're making the comparison between the risk and the benefits, and sure should be the benefits more than the risk. And also it's important to establish the dose response relationship. Example, adequate and well-controlled study to establish the efficacy should be adequate, and we know that the number it will be 1,000 to the 30,000 of the population, so it should be adequate. And randomized parallel dose response studies, because we discussed that before. Clinical safety studies of mortality outcome, large sample trials, because we wanted to know the variance, and comparative studies. These are the example of therapeutic confirmatory. And then the therapeutic use that we do it to the real population. Objective is a defined understanding of the benefit and the risk relationship, but it happens in a general or a special population and the environment. Identify the less common adverse reaction, refine dosing recommendations, because when you're just making it to the public, maybe you will discover because the people, they are not similar to each other and there is many difference between them, and maybe we just discover some side effects or some effect or something like that. And the study example: comparative effectiveness, a study of mortality, morbidity outcome, studies of additional endpoints, large samples, trials, pharmacoeconomic studies, and these things. So we discuss all the phases. The phase 1 is therapeutic use, the phase 1 is a human pharmacology, phase 2 therapeutic exploratory, phase 3 is therapeutic confirmatory, and phase 4 is therapeutic use. So we will discuss what is a phase 1 again, but in other things. The primary person is determine the pharmacokinetics in a human, because we use it in the volunteers to assess safe and tolerated dose. It's important to know that, and to detect the effects that are unrelated to the expected action. Like we do the expected action and there is something events that is happen or effects that has happened not expected, and to detect any predictable toxicity. And that's so important that we make sure what are just we determine and we just discover these things. So study classifications: pharmacokinetic in renal and hepatic impaired patients, because we know the elimination, because most of the elimination of the drug that is happening in a kidney and also the liver. Safety tolerability studies: drug interaction and food effect. We study also the bioequivalence study. Okay, so these are the study classification. And then the intervention model, because we do intervention, and the intervention, it will be the single arm or parallel, or a crossover, and we discuss these things. We said that the single arm, that's mean all the group members, we have one group and all the group members receive the treatment, or we can use the parallel, we have two groups, okay, and each one receive the treatment, or maybe case control, and a crossover, like we making an exchange. Allocation: randomized, maybe non-randomized. Okay, control: it's uncontrolled, there is no control group. If it's single arm or parallel or crossover or whatever, there is no control group. Masking: no blinding, everyone know everything. Okay, and the enrollment, we have the small number of the health of subjects. Again, I said 100 to the 500, and they are a small number really. And then we go to the phase 2, which we called it is exploratory, or we can say therapeutic exploratory. The primary person is to determine if the new drug have any beneficial activity and thus worthy. Sorry for that. So to determine if a new drug has any beneficial activity and thus worthy for of the further testing, investment of the resources, treatment, prevention, and diagnostic. Why we said the beneficial activity? Because we do this drug, or we give this drug actually to the patient participants, and we wanted to know if that really has effect or not. And the study classification, the proof of the concept, first in the patient, efficacy or mechanistic studies, dose range exploration, and pilot studies. The intervention that we use it actually crossover, and we use also the parallel design. We can do the single arm because we want to make just a comparator, because we know that if the new drug, we compare it to the existing one maybe, and so we don't have the single arm, we have the parallel, and we have also the crossover design. Allocation: placebo or standard drug. Allocation actually to the study if it's randomized or non-randomized, but actually both we can use it. And the control, we can use now the placebo or the standard drug. The masking, yeah, it maybe happened masking because we wanted to know real effects of that. And actually the masking, it's a single blinding or double blinding. Okay. um and here we use it 20 to the 500 of the patients, 100 to the 500 of the patients. So I'm sorry, the enrollment here, the small number of the health subjects, actually we use to the 10 patient, 10 to the 100 patients. But in the phase 2 we use 100 to the 500 or 20 to the 500 because the numbers are difference between many sources. And then productive classes, because it just discusses the two phases. We have the productive classes. The products may be medicinal product, drugs, or maybe medical devices, and the use for the prevention and the treatment of the disease. Yes, the drugs uses for this and the device uses for this. Mode of the action, it's pharmacological, immunological, or metabolic. The medical device is physical, sure. So the medical device is physical, and the medical products, drugs, it has two action. It may be pharmacological, it may be immunological, or maybe it's metabolic. And now we talked about the recognition of clinical studies because they are so important to discuss them, because if we do not discuss them, then this is not the lectures of the clinical trials, because the recognition is so important of the clinical studies. The first one we will discuss the global global, that's mean all over the world. For the medicinal products, I mean here the drugs, we have the Declarations of Helsinki, and we have ICH, International Council for Harmonization of Technical Requirement for Pharmaceuticals for Human Use. So both of them, they are global and they are related to the medicinal products. So we have Declaration of Helsinki and we have ICH, International Council for Harmonizations of Technical Requirements for Pharmaceuticals for Human Use. And for medicinal devices, again, Declaration of Helsinki, but we have something is called ISO 14155 2020. These are related for the medicinal devices. So again, the regulation of a clinical study that is related to global and related to the medicinal device Declaration of Helsinki, and ICH for medicinal device Declaration of Helsinki, and also ISO 14155 2020. But the regional, I mean here the European Union, that is related to the European Union, the regulation of the clinical trials for the medicinal products is called the Clinical Trial Directives and regulation and directive guidelines like 2020 EC. Okay, and we have the regulation AU number 536 2014. Again, that is related to the medicinal products, and that is regional, that is related to the European nations, all the European. So we call them clinical trial directives, and the most common is 2001-20 EC, and the other one is 536 2014. And for medicinal device, it's called medical device regulation. It's also here medical device regulation, but the numbers of it 2017 745, and unfortunately we have to memorize these things. Okay, so the regulation that is related to the Europe, we have two things. We have the medical devices and medical products. For the products, we said the guidelines 2020 AC, and we have 536 2014. For the medical devices, MDR 2017 745. These are that related to the medical device, and you can memorize it by the medical device regulation medical device regulation, and that is happened in 2017, and the number 745, I have no idea why they called it 745. And for the medical product, I don't know actually what is the 536 to the 2014, what that mean. And now we go to the national, and that is in the United States alone. There is something is called Code of the Federal Regulation, CFR, Code of the Federal Regulation, CFR, Code of the Federal Regulation. The clinical trial to be registered at the clinicaltrials.government, phase 2 to the phase 4 interventional clinical trials, phase 1 trials with the patients, actually we said to the healthy one, registry up to the 21 days after the first enrollment, results 12 months after the last primary endpoint data is collected. That's exactly what is happening in the Code of the Federal Regulation, CFR. So when you hear CFR, it's the Code of the Federal Regulation, and that is related only to the United States, and they just do that. Is like special to the United States, you know. So they have to register in a clinical trial.government, and they said that the phase 2 to the phase 4, it's interventional clinical trials. Phase 1 trials to the patients, which is opposite that we can say that, because we said that the phase 1 is related to the healthy one, and registers is up to the 21 days after the first enrollment, and the results it will be 12 months after the last primary endpoint data is collected. The regulation that is present in Germany, we can say national also, the medicinal products and the medicinal devices in the medicinal products is called AMG, Arzneimittel Gesetz. Arznei metal, it's actually one word, and Arzneimittel that's mean drugs, in Gesetz that's mean it's a rule or something like that. Okay, so AMG, it's related to the medicinal product, and we can say Arzneimittel Gesetz. And we have also the GCP for ordering good clinical practice, because we know that just GCP, good clinical for ordering, that's mean regulation. So that is related to the medicinal products. But the medicinal devices, we said Medizintechnikgesetz, medicinal Technikgesetz. End of the study, again I wanted to make sure about these things. Medicinal product is called AMG, and that's mean Arzneimittel Gesetz. And we have GCP for ordering, GCPV, which is a good clinical practice. And we have also medicinal devices is called Medical Technique Gesetz. I don't know why I called it, it's MPG, but it's actually Medical Technique Gesetz. The end of the study, notify component, federal and local authorities and gazettes, report results after 1 year. In cases, thank you so much for listening.